I'm going to be discussing weak acid equilibria in the context of a problem from Horton's 5th edition. And so let me write that. This is Horton's 5th edition, problem 2.7. And this concerns weak acid equilibria. You have probably studied weak acid equilibria, had problems on it in a general chemistry course in high school or in university. And this describes what happens when an acid ionizes. A proton is released, and the conjugate base now exists. And strong acids are called strong because they ionize completely, and we can calculate the hydrogen ion concentration quite simply just by how much acid we add. If this were hydrochloric acid, we would say what's the concentration of hydrogen ion. It's simply how much how much hydrochloric acid we added. But in the case of a weak acid, the ionization is incomplete. Many biochemical species are weak acids, and we need to have a good grasp of this concept of weak acid equilibria and pK so we can understand what charges exist and how enzyme mechanisms work. So we see several things here. We can talk about this being governed by a constant. That would be the Ka, and the Ka would be equal to the concentration of hydrogen ion times the concentration of the conjugate base times the concentration of the acidic species. Notice this is like an equilibrium constant, but these are almost always in equilibrium. They're very rapid. So another interesting idea is that we simplify this number, which can range over a very wide range, and we have it in a log form to avoid such large numbers, and we talk about the negative log 10 of the hydrogen of the Ka being we call this a pKa. Likewise, you may remember that the hydrogen ion concentration is what we're often interested in, and we have a similar symbol pH to represent the negative log 10 of the hydrogen ion concentration, and that's often what we care about. Okay, in this question we have a lactic acid, so lactic acid is shown here, unionized. Lactic acid, 0.25 molar, I would say 250 millimolar, and that is going to exist in equilibrium, very rapid equilibrium. Proton transfers are not something we have to worry about the rates usually until we get into details of high-speed kinetics, so for our purposes it's instantaneous. So lactic acid ionizes, and we can force the equilibrium of this to be a certain value by adding lactic acid as a pure compound and sodium or potassium, perhaps lactate, as another pure compound. The first part of the question is, if we mix a solution of lactic acid, that's 250 millimolar, with a solution of some lactate that is 150 millimolar, what's the concentration of the buffer? And this question is not a calculation. This question is about understanding what do we mean by concentration of buffer. And what a biochemist means by concentration of the buffer is what's the total concentration of the species that's ionized or is ionizable. And so it's simply here the sum of the acid and concentration of acid and its conjugate base. And in that case, that's easy. 0.25 plus 0.15, that's equal to 0.4 molar. So 400 millimolar. So it's a 400 millimolar lactic acid buffer. And someone might also say it's a 400 millimolar lactate buffer. There's a variety of ways to say it. It means the same thing. I would tend to say a 400 millimolar lactate buffer. The second part of the question is, what is the pH? Well, we know the Henderson-Hasselbalch equation, and this is pH, that is what we're interested in, what's the concentration of hydrogen ion, is equal to the pKa plus log base 10 of the conjugate base, that is the ionized form, divided by the concentration of the unionized form, the acid. So conjugate base and acid is what I would say. Well, that's fine. So in order to determine this, we're interested in that. We know the pKa. We were told that it is, in the question, it says the pKa of lactic acid is 3.9. So that's up here. We talk about Ka and pKa. In this case, pKa is equal to 3.9. So if we do this, we're going to have pKa is going to be, I'm going to put it below it, and we have to calculate the log 10 of this. So that's fine. We need to say, we know the top number here is 0.15. The bottom number is 0.25. If we divide 0.15 by 0.25 and take the log of it, let's look at that. What do we get? I bring up the calculator, and 0.15 divided by 0.25 equals, notice it doesn't matter if I have 0.15, 0.25, or just 15 divided by 25. It's the ratio that matters, which means that the pH of this solution, of this buffer, is not dependent in this equation by the concentrations, total concentration, but by the ratio of conjugate base to acid. And we want the log 10 in this calculator. Log base 10 is called log, natural log is ln, and that's negative 0.22 something, so let's just keep it 22. So I can write that here. So this part here is going to be negative 0.22. So that means the pH is going to be 3.9 minus 0.22, subtract 22. So that's going to be 3.68. That's the pH of this buffer. And that's the point there. If we want to make a buffer at a specific pH, that is, if we want to stabilize the pH of a solution for whatever purpose, maybe it happens naturally, like in the case of blood, or we want to stabilize it in the case of an intravenous solution or some chemical reaction we're studying in the lab. The way we do it is by we find a buffer system whose pKa is close to the pH we want, and then we mix the acid and the conjugate base in the appropriate ratio to get the pH, and we have to calculate it this way. This is a very common thing to do in the laboratory. And sometimes we will have in biochemical practical manuals lists of appropriate buffers and charts that show for any specific pH with that acid-base combination what ratio of molarity should be used. So what does this mean? This also means that if let's look at let me put an axis here representing pH. So if this is an axis representing pH, and we have something very alkaline here, and we have something very acidic here, and 1 to 14 is a pretty good representation of anything we're likely to see biologically. So we have something very acidic here, and we have something very basic or alkaline here. What's the pKa mean? Well, the pKa not only will tell us what pH how the pH can be controlled, but it's also useful that if somewhere in the middle here is pKa, that means for a species like this, the acid form is going to exist, in that this lactate at any pH below its pKa will be protonated, and any pH above its pKa will exist as its conjugate base. So that's very useful, because very often we're interested in an amino acid side chain, for example, and we'd like to know, is it charged or is it uncharged? How do we know whether it's charged? Well, if we know the pH and we know the pKa, we can say at this pH, this molecule is charged, this part of the molecule is charged, or it's uncharged. And if we're looking at a base, then we talk about a base, and we talk about a base, let me show that differently. We can talk about a base as being something that accepts a proton, and we usually, when we discuss pKa's, we don't talk about pKb's. Instead, when we talk about the pKa of a base, we're talking about the pKa of its conjugate acid. So if this were ammonia, ammonium plus H+, it's an equilibrium with ammonium ion, and we'll talk about the pH or pKa of ammonia. What we really mean is the pKa of the acid form. And likewise for a base then, if the pH is more acidic, it's protonated, it exists in the protonated form, whether that's, in this case, it's going to be positively charged, and above the pH, it's going to exist in this neutral form. So that's very useful for us conceptually to say, is this molecule, is this side chain, is this part of the molecule charged? Is it negative? Is it positive? Is it neutral? How do we know that? We know that by looking at knowing the pH, and then knowing its pKa, and then realizing at this pH, these parts of the molecule are negatively charged, these parts are positively charged. This is very important later when we get to discussion of enzymes.